that Dr. Elizabeth Rowe is here today to talk about her research. Elizabeth and I have known each other for, I think, like 10 years.
Yeah, something
like
like that.
that. So
we go back.
We go back. We've known each other since we were grad students. And Elizabeth and I met over our mutual admiration for the endometrium, which I have to admit is not an admiration that many other people in the world share. So it was especially exciting to meet somebody else who was interested in this very cool tissue. Elizabeth got her PhD in 2011 from Temple University. Her advisor was Dr. Christy Rockwell. She did some really cool work on the progens and endometrial thickness. Elizabeth now has a position at Purdue in anthropology and teaches work that intersects really nicely with evolutionary anthropology, and nutritional sciences. So, without further ado, I will let Dr. Rowe tell you more.
Thank you for that lovely introduction, and thank you for inviting me to what feels like a sister campus to Purdue, although this is a nicer place, I've got to say, a nicer campus here. It's prettier. Okay, so this title is a bit of a mouthful, right? Ancestral resilience, evolutionary significance, et cetera, et cetera. Really what I'm interested in is how our ability to cope with changes to our environment, how that's embedded in our genes, and how evolution may have embedded those changes into our genes that make us more resilient or able to cope with environmental changes. And then looking at today's life, trying to understand what consequences that might have for health, okay? And really what I'm looking at is, it's all related to women's reproductive health, but this kind of methodology, this kind of theoretical perspective is applicable to really any aspect of human everyday life, our ability to cope with stress and deal with stress. And in fact, the people who are most interested in this kind of work have really looked at how this impacts people's ability to handle stress, okay? But so it's not just about women's reproduction, it's about everything, right? But so to kind of give some context for my work, in terms of what I'll be talking about today, I'm going to be talking about what we know theoretically about how women's reproduction has been shaped by evolution, especially by its ecological circumstances. So in other words, by the environment in which women find themselves, how that's been, how our ability to reproduce has been shaped by that in particular environment. How it is that we think genes might be able to shape our ecological response. In other words, how can genes shape our ability to cope with changes in the ecology, okay? And how, looking specifically at women's reproductive ecology, what do we think might be going on there in terms of genetic variants shaping our ability, women's ability to, the ability of women's reproductive systems to cope with changes to the environment, okay? And of course, we'll talk about the anthropological significance of my work as well as the broader significance, especially related to women's health. Okay? So I want to talk about, just in very broad terms, what we know, or what's been really dominant in the field of anthropology with regard to how women's reproduction is shaped by the environment. So we understand pretty well, we've understood for several decades, if not even longer than that women's reproduction is very sensitive to energetic constraints. Your average person certainly is aware that women who are elite athletes often, they might not have their first period until they're relatively old compared to the rest of their age cohort. They might have fewer periods and so forth. So for a long time, we've understood that there's some, at least with regard to the menstrual cycle, there's some aspects of the energetic environment that seems to be shaping that. So in this case, of course, we know these women, even if they're well nourished, they have high levels of physical activity, and that seems to be in some ways kind of down playing their reproductive system, okay? Well, from an anthropological or evolutionary perspective, we can actually explain that using evolution, natural selection, right? So you would think, okay, the whole point of natural selection is to reproduce, to make babies, so stopping reproduction is sort of antithetical to that. Of course, we understand that with humans, pregnancy is very energetically expensive, right? We have this large brain in our fetuses that we are trying to give birth to. That brain is incredibly calorically expensive, on top of women trying to keep their own bodies going. So the idea is that we're actually shutting down reproduction if energetic times are not so great. If we have a high level of physical activity, if nutrition is hard to come by, if nutritious food is hard to come by,
the idea is that really, it may be good to wait until energetic times, energetic circumstances improve, okay? And that way we can maybe have a better quality pregnancy, because the human way of dealing with natural selection seems to be not having so many babies, but having really high quality babies, 1 or 2 or 3 or 4 or 5 high quality babies over a lifespan, okay? Unfortunately though, when we talk about fertility, fertility is really the number of offspring that you have that survive to adulthood, right? And that's really hard to measure over the human lifespan because we live so long, right? And we live so long relative to ourselves, so it's hard to study ourselves from that perspective. So instead, what anthropologists and other people who are interested in women's reproduction have often studied is something we can refer to as fecundity. So fecundity, just broadly understood, is the probability of you becoming fertile or becoming pregnant with any given menstrual cycle. And there's a number of measures that we can use to actually look at that, okay? So we can look at things that happen over the menstrual cycle. So this is, of course, what sort of the lining of the uterus is doing over the menstrual cycle. It's building up and it's being shed, and we can measure that. We can maybe measure menstrual bleeding. We can look at 2 of the dominant hormones produced by the ovaries as sort of an indication of what's going on in the ovaries and how prepared the ovaries are for pregnancy. We can look at estrogen and progesterone, okay? So this is typically what people in our field have done. In the past couple of decades, what anthropologists have done actually is looked at, aside from these Western populations of women who are these high-level athletes who are well-nourished, let's look at other populations that live more of what we might consider to be a traditional lifestyle. Folks that whose lifestyles might better represent what was going on in our evolutionary or ancestral past. Okay? So in these populations, these women, their nutrition may be very seasonal. That might vary with regard to harvest seasons or planting seasons. Their workload also varies pretty strongly as well. So they might have high levels of workload during planting seasons or during harvest seasons and otherwise. Okay, so these are women in Nepal threshing grain. Okay, and when they've looked at measures of fecundity in these different populations around the world, what they've noticed is that women in these populations that live more of a traditional lifestyle, they have these heavy workloads, or they may have more seasonal variation in nutrition, they tend to produce very low levels of hormones. This is a hormone progesterone. And this is sort of an idealized representation, and it's average, and no woman actually looks like this, right? But what we see is that the idea is as sort of the beginning of the menstrual cycle, and this is actually when a woman starts her period, the low, levels of progesterone are quite low, and then you see this little hump after ovulation roughly there, okay? So these are women, these are Nepali women, right? We don't see much of a hump with those women there in terms of their progesterone levels. Compared to women in Boston, though, these women in Boston tend to produce high levels of progesterone. So well-nourished Western women produce high levels of this hormone, okay? This has been a major contribution by our field to the understanding of women's reproductive biology. Okay, so the idea here is that just high energetic stress lowers fecundity, and that we think that the idea, at least theoretically, is that if you have this high level of energetic stress, you have high levels of physical activity, low levels of nutrition, it should somehow impact your fertility over your lifespan. It should somehow constrain your fertility. Okay? So this is kind of the model that has been predominant, one of the predominant models in our field, okay, where you have fecundity kind of performing this balancing act between these 2 extremes of nutritional stress, right? Where you have a high workload, you have high energetic stress, you have a high workload, maybe poor nutrition, and maybe you don't have a lot in terms of fat stores, you don't have a lot of energy stored up in your body. That has maybe a downgrading effect on fecundity, right? It lowers your fertility. Whereas if you have lots of energy available, you have a low workload, you have lots of available food, you have high fat stores, okay? That tends to make it more likely that you're going to become pregnant in a given cycle. Okay, so this is a dominant evolutionary model that's out there, okay? The problem is this model makes some assumptions about our ancestral past. So this is the Juhuasi of the Kalahari Desert, the kind of favorite anthropological model for understanding our ancestral past as hunter-gatherers. And of course, as we've gotten a more sophisticated understanding of what our ancestors were doing in the past, these folks are probably not a great model for what our ancestors were up to. Nevertheless, we still kind of talk about them as if they were. So when we look at these folks, the kind of understanding is these folks have a very high workload, you know, in terms of their everyday life. The understanding is that they go out every day and they forage. Women are foraging, men are hunting. So they have a relatively high level of physical activity. They're not, you know, endurance athletes by any stretch of the imagination, but they are definitely very physically active. And the idea is that they're somewhat constrained energetically, right? If these folks live in the Kalahari Desert, they must not be getting a whole lot of food, right? That's the idea. So this kind of evolutionary model here is kind of saying, you know, these folks are kind of living in an environment where they're kind of on the cusp here, right? And if things just get a little worse, the idea is that that's going to really constrain their fecundity and make it so that they're less likely to become pregnant. Of course, we know this model of understanding that these folks are sort of constantly on the precipice of becoming nutritionally inadequate is probably not correct, because of course these folks, whenever nutrition gets bad, times get tough, what do? They pick up and move, right? They don't stick around in an environment that's really unpleasant to be in, okay? So this is a model that's making this assumption that this is how our ancestors were living. And it also sort of, the idea here is also these folks represent us genetically, that we haven't changed in the past 10,000 years since humans got involved in food production, okay? So in fact, there may be more than one way, at least I would say at least two different ways of understanding how women's fecundity can respond to energetic stress. And I'll talk about exactly what I think may be mechanistically going on in terms of how our bodies are able to kind of discern, you know, what's going on. And these two pictures are, of course, a woman milking a yak and women involved in rice planting, two of the major food-producing activities that women around the world are involved in contemporary populations where they have animal herds and where they're engaged in hand labor in terms of planting. Okay, so this is, of course, our model that we've been talking about here, all right? So what this model is essentially saying is we have this fecundity, you know, being downgraded and when times are tough. This model is sort of saying that all women should be universally the same in terms of their response to energetic stress. So when times get tough, there's lots of physical activity going on, they're engaged in harvesting, there's not a lot of food, their fecundity should go down, okay? So we might say, in fact, that what if only some women are actually sensitive in that way? What if only some of the women in a given population are actually sensitive? And in fact, what if some women actually don't respond in that way at all? What if they respond in either the opposite way, or if they nothing really changes for them, right? If the environment gets bad, maybe things don't change so much for them. Let's say maybe these women are what we could call resilient. They're resilient to these changes in the environment. Okay? So what we might actually see is some women who are sensitive, some women who are resilient, and we might actually look for genetic variants that can account for these differences in sensitivity. Okay? So that's really what my work has been about thus far. So I want to talk about how it is that genes can actually shape our ability to respond to the environment. So when we talk about genes, we often understand the genetic variation in terms of how, you know, that can affect things in terms of protein shapes, right, amino acids and all of that. But actually some of the more interesting genetic variation in the human genome actually relates to gene expression. Okay, and this picture I think nicely encapsulates that. We have a man and a woman here, and you can see that there's pretty clear differences in terms of how these people can respond to the ecological stressor of sunlight, right? Clearly, one of them has either had more exposure to sunlight or has a genetic predisposition to producing more melanin in his skin compared to the woman, okay? So there might be some genetic variation between these two, aside from their differences in sex chromosomes, that could account for this variation you see in skin color with exposure to sunlight, okay? And of course, we do know that there's genetics involved in that, okay? So what's going on maybe at the molecular level, okay? We have tons of genes in our body, something like 20,000 genes. If we had all of them turn on in our body at the same time, we would be not functional. Bacteria don't even do that, okay? So what we actually have are these switches that allow genes to be turned on and turned off, okay? So these are actually things called, well, there's a whole bunch of stuff involved here, but one of the things that's involved are called transcription factors, right? And what transcription factors do, and that's what this guy is here, these guys look for, they're essentially molecular signals that look for particular DNA sequences that are not actually part of the gene that's being expressed, but often nearby or sort of in front of it, okay? So they recognize these specific DNA sequences, right? And if there's variation in these sequences, and this is called something called a TA box, so this guy is looking for this particular DNA sequence. When it sees this, it says, okay, I'm going to latch on here, I'm going to turn this gene on. Well, what if you have, another area later on in the gene where you have this exact same sequence, okay? That means this gene has the capacity to be turned on twice as often, right? So it's sort of like an additional switch. And conversely, you could have something that could basically alter this sequence so this is no longer recognized by this transcription factor, so the switch is no longer turned on. So you have things that can easily turn on or turn off that are embedded in the actual sequence of the DNA, okay? So, essentially what we're talking about is variation in these DNA sequences, so variation at the genetic level that can alter, that have the capacity to alter our ability to respond to these molecular signals, okay? So you have a molecular signal, some kind of thing that tells your body, okay, you're being exposed to this amount of sunlight, and if you have an extra switch that allows you to turn on extra melanin production, that might account for why some people have darker skin when they're exposed to sunlight. I always feel like I have like a big farmer's tan after the summer like we've had, where it's very bright and sunny. And clearly I have some capacity for producing extra melanin on my skin. Okay. So, if we kind of relate this back to what we've been talking about in terms of women's resilience or sensitivity, these gene switches can then explain this variation that we see in sensitivity and resilience. Okay. So I want to talk about how it is that the genes can shape women's reproductive ecology. Okay. So this is kind of what I've been talking about here, that there might be some women who are resilient and some women who are sensitive to energetic stress. Okay. So how could that happen? All right. So in order for your body, the things that we actually measure with regard to fecundity, those things are actually often hormones or response to hormones, okay? Hormone production and response to hormones are actually, that's all governed by genes, all right? Hormones themselves are not always encoded by genes, and the ones that we're talking about here certainly are not, but our ability to respond to them, the production of these things, they're all proteins that are enzymes that make genes or proteins that make receptors for hormones, okay? So, what's interesting is that we think that these genes that either are involved in hormone production or hormone response actually are sensitive to what's going on energetically in the body. So this is a very complicated diagram. Basically what this is a cell. So this is a cell membrane, and then this is the nucleus of the cell with our DNA inside the cell, okay? What's going on here is at the surface of the cell, your cell is actually able to respond to glucose, right? So blood sugar. That's a major indicator to your body of your energetic status. How much sugar do you have available, right? Your brain and your muscle tissue are incredibly dependent on glucose for their functioning, right? And so your body is very sensitive to that, okay? When you have glucose, what this essentially does is this turns on this sort of a bunch of different little switches, like dominoes, within the cell that says, okay, now it's okay to produce, to actually respond to progesterone, okay, or to produce a hormone, okay. And this case, it's actually a receptor, okay. And what's interesting is that some of the receptors for hormones themselves are involved in actual production of genes downstream of that, okay. So let me tell you a little bit about how I actually went about studying this from a methodological perspective, okay? I wanted to study what was going on in terms of how gene-environment interactions were affecting women's reproduction in healthy women, okay? And that's really one of the things that sets anthropology apart from other disciplines when we study women's reproduction, or really any aspect of human body functions. We're often looking at things beyond just disease, and often people in biomedicine are really restricting themselves to what's healthy and what's not, okay? So I was looking at these women who were healthy. They also couldn't be pregnant or on hormonal contraceptives, and this population was based out of the university that I was working at, so a lot of these women were college age, and it is very difficult, by the way, to find women who are not using hormonal contraceptives like the pill. So at about 50 women, their ages 20 to 45, they had to be at least age 20, and they couldn't be postmenopausal either. I was looking at women who were within the healthy, normal to overweight BMI range, and so BMI is body mass index. It's an index, in other words, of your weight relative to your height. There's a lot of issues with BMI as a measurement, but part of the reason why I was using this as a criterion is that we know that women who are underweight or who are obese tend to have irregular menstrual cycles and irregular hormone production. So I was trying to avoid looking at that population. Okay. So yes, they were not using hormonal contraceptives, not pregnant, and they couldn't be nursing either. Okay. So how I actually went about doing this study, I had the women come into our laboratory and fill out a questionnaire as well as give informed consent. So of course I had IRB approval from my university for this as well. I had them basically swish some Listerine in their mouths and spit into a tube. So I got some saliva, which also contains cells from your cheeks, so I could get their DNA from that. I collected I measured their height and weight at this visit, and also measured their waist and hip circumference as well. So I collected a bunch of data about them initially. So this was the initial study visit, and then I had them pick up these bags. It's literally a grocery bag that was completely filled with this take-home kit for them. So this take-home kit actually contained pads and tampons, as well as a rack essentially of kits, of tubes to collect saliva, which I then later analyzed for saliva. So looked at, I analyzed them for hormones. This is ovulation test strips. It's kind of hard to read, but I also had them collect urine at home in a little cup, and then do their own ovulation test strips, very much like a pregnancy test. Okay. So I had them, using this material, I had them track their menstrual cycles for 3 consecutive cycles and collect data about their menstrual bleeding. So I had them use the pads and the tampons. One way to measure how much menstrual blood women are losing is to actually have them bring back the used pads and tampons, and then there's this whole process where you squeeze out the blood and all of that, and I decided I didn't really want to do that, but really more to the point, I didn't think that women would be willing to enroll in a study where they had to bring in pads and tampons to a laboratory. Yes, you know what I mean. Okay, so really what I had them do is actually use a little pictorial chart to kind of estimate how much menstrual blood they lost. So I actually have a rich data set here in terms of menstrual blood loss with this population. I also had them track their physical activity data on a calendar, so how active they were physically or how active they weren't. And as I mentioned, they collected saliva samples, which I then analyzed for hormones. They only did this during the 3rd cycle of the data collection period, and they kept these tubes in their home freezers, by the way. Okay. So, in terms of getting back to this whole idea of understanding how genes might shape our ability to respond to ecological stressors or energetic stressors, I didn't look at, I didn't really use a strong marker of physical activity. I didn't have them, carry around physical activity measurement recorders and things like that. And I didn't have them do nutrition diaries, in part because I actually suspected that hormones would have a big impact. There'd be a big interaction between hormones and genetic variation on reproduction, and that's not actually what I found. So the fact that I found some interesting, significant results looking at these kind of proxy measures of energetic stress, and here I was looking at physical activity, and I was looking at these measurements of their body, so BMI and waist and hip ratio, which is WHR. I was using these as very proximate measures, but it's very interesting that I nevertheless found some kind of significant results here. Okay, so I used kind of 2 proximate measures of fecundity. One was estrogen levels. So this is kind of showing what estrogen does over the course of a menstrual cycle. Okay, and so you can see this is relative to day of ovulation. So this is when a woman's period starts, goes for 5 to 7 days, something like that. And you see this peak in estrogen usually right before ovulation. And what's interesting is this chart shows us is that women who experience clinical pregnancies, in other words, a clinically observable pregnancy where they can do pass a pregnancy test, in other words, with these women, they tend to have higher levels of estrogen. So, in other words, having higher levels of estrogen usually indicates a more fecund cycle, more likely for a woman to become pregnant. Okay. What I found is I looked at a gene involved in production of hormones and production sort of upstream of producing estrogen. And I found that this gene alters estrogen production in relation to waist-hip ratio. So if you had this particular variant of it, you seem to be more sensitive to energetic stress, and in this case, waist-to-hip ratio. So as your waist-to-hip ratio actually increased, your estradiol production went down, and this group of women seemed actually not very sensitive at all. They were resilient, right? They didn't change in response. Okay. So what I think is going on here is that there's some indication, at least from my data, that this genetic variant alters fecundity in relation to energetic stress. Of course, this is not an energetically stressed population, okay? This is looking at women who are healthy, probably well-nourished as far as I'm aware, okay? So it would be interesting to replicate this in a population that is indeed energetically stressed. All right. So that's just one finding that I had. This other finding I wanted to mention to you relates to menstrual cycle length. So we know that menstrual cycle length is in some ways actually a measure of fecundity as well. So as you see in this population here, women are actually, in this population at least, women were more fecund if their menstrual cycles lasted from 30 to 31 days. So that's, a period starts up, okay, and then we actually say that the menstrual cycle ends when the next period starts, okay? So if you've heard of that whole 28 days thing as being a typical menstrual cycle, clearly some women might have 28-day cycles, but clearly, at least for some women, they're more fecund if their cycles are a little bit longer than that. So this is I use this as another proxy measure of fecundity here, okay? And I looked at a gene that's related to hormone response. And again, I found a very similar kind of thing where I was looking at waist-to-hip ratio, okay? And we have a gene here, I'm sorry this is cut off. We have clearly one variant of this gene involved in hormone response actually was much more sensitive. And this one group here is quite resilient. And as you can see, I've put this green line around this. It actually falls around this entire group. They all pretty much fell around this time period of 30 days or so that seems to be most fecund. So they do seem to be quite resilient to energetic stress. Okay. All right. So overall, what I think my results are showing is that hormone production and response genes can alter fecundity in response to energetic stress. And I've just shown two results. I actually have a lot more that are in some of my research papers that I based this talk on. So it's not just what I'm showing you here. I'm showing you kind of two selected pieces of data. But overall, there does seem to be some interesting interactions here. And again, mechanistically, it's quite interesting because we're talking about things that are related to response to progesterone, response to estrogen, not response to how big your body is, right? But there clearly seems to be some mechanism by which that information is being transmitted at the genetic level. Okay, so what's the anthropological significance of this? Okay. I already mentioned this model that I was talking about before, where we think that increased energetic stress tends to have a downgrading effect on fecundity. You're less likely to become pregnant when energetic times are kind of tough. Okay. We might actually kind of supplant this idea of fecundity and actually understand that insulin is actually a good marker or indicator of energy balance. So of course insulin is a hormone that's important for blood glucose regulation, okay? And interestingly enough, insulin levels kind of reflect a couple of different things, including anthropometric measurements like BMI and waist-to-hip ratio, as well as your physical activity, right? So people who have a higher BMI, a higher waist-to-hip ratio also tend to have high insulin levels, and of course the converse is true, okay? So in some ways, insulin is a marker, an indicator of your, what we might call your energy balance, how much energy you have coming in relative to how much energy you have coming out, okay? And I didn't present data to you on BMI or physical activity. I've just been talking about waist-to-hip ratio. So we might think of waist-to-hip ratio as being somewhat of a distant measurement or distant indicator of what one's insulin levels might be like. So in our population, we might say perhaps that the women who had high waist to hip ratio also had high insulin levels, and then of course women with low waist to hip ratios also had low insulin levels. Okay? So, what might be going on is that these measures of fecundity might be altered in response to actually variation in insulin, right? So I showed you data related specifically to waist to hip ratio. What might be going on is that these genetic variants are in some way sensitive to variation in insulin levels. Okay. So what does this have to do with what I was talking about earlier with foraging populations? Okay, it might actually be related to these different subsistence strategies. We actually think that pastoralist populations, women and men involved in food production, they're actually much more dependent on sugar in their diet than we think foraging populations probably are. So if you're a pastoralist, if you have animal herds, and a lot of people who have herd animals, they're not consuming the meat of the animals, unless it's pigs, because nobody drinks pig milk. Most of the time, if you have herd animals, you are directly consuming either their blood or their milk, okay? And typically from their milk, you're getting a lot of sugar as lactose. Of course, you're getting protein and calcium and other nutrients as well. But sugar is a major factor there, okay? And certainly if you're heavily dependent on food production and as agriculture, you're also getting a lot of carbohydrates, which our bodies break down very quickly into sugar, right? And we convert these sugars, lactose, into sugars from grains very easily into glucose, which then our brains use, right? And of course, this is all related to insulin. Okay, so we think that, I think maybe what's going on here is these populations might actually vary in their insulin sensitivity, okay? So foraging populations might be relatively insulin insensitive, right? Insulin is not a great indicator of energy balance for them, right? They, again, they may not be exposed really to much variation in insulin anyway, because they can pick up and move if times get tough, right? There's no sense in being terribly insulin sensitive if you're a foraging population. But if you're an agriculturalist or you're a pastoralist, if you're involved in food production, you might be more insulin sensitive simply because you're much more reliant on food sources that are rich in these carbohydrates or sugars. Okay? So there might be this variation in insulin sensitivity, and that might account for this variation that we're seeing in terms of response of fecundity markers to energetic stress. So it might be that foraging populations are insulin insensitive. In other words, they are resilient, whereas food-producing populations are insulin sensitive. Okay, so I think that's what the anthropological significance of it might be. In other words, this model that we've been talking about is maybe a little too simplistic, and actually we have at least two ways of responding to variation in insulin in our environment. Okay, so what's the broader significance of this research? Well, we widely acknowledge, it's widely understood that the environment that our ancestors lived in is pretty different from our current environment. Okay, so our ancestors, even if you think of them as foragers or food producers, they're much more physically active than we are. Probably, generally speaking, unless they were wholly dependent on a grain resource, probably getting much better nutrient profile in their diets. We think this has some consequences for chronic disease risks. So things like diabetes, but especially reproductive cancers that seem to be sensitive to lifestyle changes. Okay, so interestingly enough, we certainly understand that cancers like breast cancer are absolutely, there's actually a very strong genetic component to cancer risk. Right? We all know that there's hereditary cancers. Breast cancer can be hereditary. Okay, people vary in terms of their overall load of the amount of variants that they carry in their bodies that can actually increase their cancer risk. Right? So some people have a very low level amount of genes that can increase their cancer risk. Some people have very high level. But in a population that is not actually very conducive to causing cancer, not very many people in this population are gonna get cancer. So if you're living in an environment where you have great nutrition, you have great levels of physical activity, even if you have a high load of risky genetic variants that might give you cancer, you're at lower risk for getting cancer, okay? If, on the other hand, you live in a cancer-promoting environment, right, as we kind of do, where you're exposed to all kinds of environmental contaminants, you're exposed to overnutrition and a low level of physical activity, and again, we know that some cancers are very, very sensitive to that. If you live in this cancer-promoting environment and you have a high load of these risky genetic variants, you are a very vulnerable person. And of course, we know that this is a whole population of people, it's not just one or two individuals, right? So these people carry a high load of these genetic risk variants for cancer, but they're also living in our contemporary, very high risky environment for cancer that tends to promote cancer. Okay, what does this have to do with, what we can do about cancer in terms of cancer prevention and that kind of thing? I wanted to point out that, not only do we live in an environment these days where we live in an environment that tends to promote cancer. That's not true for all of us. There's a high level of variation in terms of our cancer risk between individuals of differing socioeconomic status. So women who are food insecure actually are at a higher risk of obesity than men who actually face insecurity in the United States. So food insecurity, by the way, is not just do you have enough food to eat, but it's also things like, do you know where your next meal is coming from? Do you have to make budgetary changes to pay your rent versus buy groceries? If you have kids, maybe you're going to let them have dinner and you're going to go hungry. You would think that food insecurity would make you skinny. It turns out that among women, again, in the United States, being food insecure, they're much more likely to be obese. Okay? So what we're looking at is a population that is especially they're especially at risk because of this environment that is likely to promote cancer in this population. So these women, among those women who have a high load of genetic risk variants for cancer, if they're also food insecure, they are also incredibly vulnerable to cancer. So it's not as if we can't do anything about these kinds of things. You can't do anything about the genes that you carry, especially if there are thousands of the genetic variants that predispose you to cancer. But we can do something to alleviate the problems like food insecurity that some people face that actually make them incredibly vulnerable to cancer. Okay, so that is my talk. All right, I wanted to acknowledge my funding agencies, Wennergren and the National Science Foundation, as well as Temple University, and my collaborators at Temple University and at Temple Medical School as well. So I'd be happy to take any questions that you have about my research. Yeah. Yeah.
I wonder, one of the things that I thought was kind of interesting about your talk is some of the variables, when you're talking about your own research, some of the variables that you're drawing from are related to energy availability, like high or low physical activity, but some of them are more related to things like energy status or energy stores, like waist to hip ratio. Mm-hmm. So can you talk about the ways in which not just the amount of insulin in your body or your energy availability, but also your fat storage, like as another marker of energetic status? Like, do you feel like those could have different effects on your model, or?
Yeah. So actually, I was just talking with Dr. Taryn Garcia about maybe collecting some leptin data. Yeah, right. So we don't really know. So leptin is actually an indicator of fat stores, roughly. So that would be an interesting thing to look at. From my perspective, though, I mean, the insulin thing really stood out because of the waist to hip ratio difference. And I don't think you see that correlation between waist to hip ratio and insulin that you see. I don't think you see that between leptin and waist to hip ratio. I don't think that's right.
Depends, because waist to hip ratio is tricky, right? Because what it's also telling you, sorry, I'm not sure who does that.
Yeah.
What it's telling you is just the relationship between these two.
Yep.
So for some people, that can be a lot of that, and other people, that can be less adiposity. So
yeah, and it's an incredibly crude measure too. I mean, I actually have some really interesting, I don't think you've seen this before, that kind of principal component analysis, where it's not just taken into like account like the ratio between those two things, but the actual measures. So yeah, there's there's a thing, there's something going on there too. Yeah. Good question. Yeah. Yeah.
Have you, do you have any data on insulin levels in
No.
these hunter-gatherers?
No.
So that's just a hypothesis.
It's just hypothetical, and, you know, again, this population was very well nourished, so it would be interesting to be able to replicate that with actual insulin or insulin biomarker data in that kind of population.
Some of the research that I know about seasonal nutrition among the Kalahari hunter-gatherers collected back in the 70s and 80s
Yeah.
before they had access to external food aid. Shows that they had highly seasonal breeding. compared
Yeah,
to
I know
the
what you're talking about, yeah.
pastoralists, next to them. What does that say about what insulin levels would have been?
Well, I mean, you know, if my model is correct, what that's saying is maybe that actually in some populations of foraging populations there is seasonal variation. So it may not be whether or not you're a food producer or a forager, but whether or not you know, there's seasonal variation in food production. I know in that population they're heavily dependent on mongongo nuts, so I don't know if there's seasonal variation in that. With regard to the dry season and the wet season, I don't know.
Well, we can look back at the data.
I know, the
Take
thing is,
fairly
it's again, it's,
comprehensively measured in that study, and seasonality. It was in comparison to the adjacent agriculturalists that
Right.
had much less seasonality of birth that I found interesting.
yeah, yeah, okay, good point. Other questions? Yeah.
So, have there been any studies in diabetic women or women who have a lot of fluctuation in insulin and how that relates to fecundity?
Not as far as I'm aware, and one of the problems with that is that there's often the comorbidity of polycystic ovarian syndrome with women who have diabetes or have insulin resistance. So often, you know, looking at things like estrogen as a biomarker of fecundity may not be really relevant because they're often very flat in terms of their estrogen production because they're not ovulating. Good point, that's a good question. Yeah.
Following up from that, did you look at like any models of ovulation, or you had the like LH strips to test for ovulation in terms of that as a marker of fecundity, potential fecundity, in relationship to these?
Yeah, so I just
Anybody?
used the ovulation test strips to kind of, I was actually just using it to pinpoint ovulation with this population because I was trying to time the measurement of the ultrasounds I was doing for endometrial thickness.
Okay.
So it was a completely different thing, but you know, actually most of the women in my population, they were ovulating, either as indicated by that. I mean, I only had them check that over a limited number of days, but I also have estrogen and progesterone data that does indicate that most of them were actually in fact ovulating. Yeah.
Did you remove the anovulatory cycles then from the analysis, or did you compare everybody together?
You know, in one of them I actually did with the one where estrogen production is involved. Yeah, so we did that, yeah. Good question. Any questions I didn't answer on the issues that you wanted to find?
You got them all.
Good. Yeah.
Could you talk about how your ideas relate to the paper by Wells in Current Anthropology about fat storage and its relationship to reproduction and intra-annual variability in environments? He calls this the capital breeding strategy.
I'm not familiar with that paper.
Okay. It's worth looking at. 'Cause it's
Mhm.
about
When was it
fat
published?
storage, December 2012. It's called The Capital Economy and Hominin Evolution. And the subtitle is How Adipose Tissue and Social Relationships Confer Phenotypic Flexibility and Resilience in Stochastic Environments.
That sounds like it's really familiar, very similar to the work that Claudia Valletti has been doing, looking at essentially how people extend energy to other members of their social group in terms of reproduction.
Part of it, I think.
I don't read current anthropology very often.